Cell signaling is basically the biological version of texting, except cells don't ghost each other as often. Every moment, your cells are whispering biochemical secrets to one another to keep you alive. They pass messages in the form of molecules, ensuring no part of your body forgets what it's supposed to be doing. Without signaling, your muscles wouldn't contract, your heart wouldn't beat, and you'd probably still be asleep somewhere. The signaling process follows a logical three-step pattern that even the most chaotic cell must obey. Each signal molecule has a perfect receptor buddy waiting to bind with it like a molecular dating app. When signaling goes wrong, cells panic, behave badly, and sometimes start forming tumors just to spite you. Cells don't always yell. Sometimes they whisper politely to their neighbors. In local signaling, cells release molecules that drift short distances, like biochemical perfume clouds. Neurons take this to the next level by launching neurotransmitters across synapses in a dramatic chemical leap. Long-distance signaling, however, is the hormonal equivalent of sending a message through the postal service. Hormones travel through the bloodstream to faraway target cells that actually care about the message. Cells without the right receptors completely ignore the hormones, like their spam emails. The distance a signal travels determines how quickly the whole system responds, making cell communication a bizarre mix of speed dating and snail mail. Cell signaling has three elegant stages, like a biochemical three-act play. The first step, reception, is where the signal molecule, called a ligand, grabs onto a receptor, like it's shaking hands with great enthusiasm. Then comes transduction, during which the cell converts that handshake into a series of biochemical fireworks. This stage often amplifies the original message until it's louder than a megaphone at a rock concert. Finally, the response stage guides the cell into actually doing something useful, like dividing, secreting a protein, or chilling out. These three stages are used by everything from bacteria to humans, providing or proving biology loves reusing templates. If any stage fails, the resulting confusion can spark disease, chaos, or mildly annoyed biologists. Receptors are like security guards stationed around the cell, checking every incoming ligand's ID. Each receptor only binds to very specific signal molecules, much like a picky bouncer. When a ligand fits perfectly, the receptor changes shape as dramatically as a soap opera actor in mid-scene. This structural shift triggers downstream events inside the cell that launch the signal transduction pathway. Some receptors sit politely on the cell membrane, while others lurk inside the cytoplasm, waiting for lipid-soluble signals. A mutation in a receptor can make it overactive, underactive, or just plain weird. Evolution tweaks receptors constantly to help cells survive whatever biochemical nonsense life throws at them. GPCRs are like the rock stars of the receptor world, showing up everywhere and and attracting enormous scientific attention. When a ligand binds a GPCR, it wakes up a nearby G protein that's been loitering on the membrane. This G protein then darts off to activate enzymes like it's late for a biochemical appointment. Those enzymes produce second messengers, tiny molecules that spread the word inside the cell. GPCRs help you smell cookies, taste pizza, and see colors, so they're basically responsible for life's joys. Many medications target GPCRs because controlling them gives you major power over cell behavior. If GPCRs ever went on strike, human life would get very inconvenient very fast. Receptor tyrosine kinases are the overachievers of cell signaling, always multitasking and always phosphorylating. When a ligand binds, two RTK monomers snap together into a dimer like molecular BFFs. This activates their kinase regions, which start slapping phosphate groups onto tyrosines with great enthusiasm. These phosphorylated sites become docking stations for relay proteins that launch various pathways. RTKs are heavily involved in regulating growth, making them both essential and extremely dangerous when they malfunction. Many cancers involve out-of-control RTKs that refuse to calm down. If GPCRs are the rock stars, RTKs are the ambitious CEOs of the cellular world. Secondary messengers are tiny molecular interns running around the cell, spreading information at lightning speed. CAMP is one of the most famous, happily bouncing around activating protein kinases. Calcium ions are also major players, causing everything from muscle contraction to neurotransmitter release. IP3 acts like a biochemical locksmith, opening calcium channels in the endoplasmic reticulum. Because second messengers move so fast, cells can respond almost immediately to signals. Their small size allows massive amplifications. One receptor can lead to thousands of molecules racing around. Without second messengers, cells would respond to signals about as quickly as a sloth solving a math problem. Signal amplification makes cells incredibly efficient at detecting even tiny amounts of signaling molecules. A single ligand binding one receptor can unleash a biochemical chain reaction worthy of an action movie. Enzymes activate multiple targets, which activate more molecules, and soon the message has exploded across the cell. This allows cells to respond to signals even when they are present in microscopic concentrations. Amplification also ensures that weak environmental clues still cause meaningful responses. However, if amplification goes unchecked, the cell can overreact in dramatic ways, something biology loves doing. Proper control ensures signaling stays helpful instead of causing cellular melodrama. Intracellular receptors are introverts that hide inside the cell, far from the noisy membrane. They wait patiently for lipid-soluble molecules like steroid hormones to wander in. These ligands glide through the membrane effortlessly because lipids love hanging out with other lipids. Once bound, the ligand-receptor complex transforms into a transcription factor with big ambitions. It enters the nucleus and directly influences gene expression, essentially rewriting the cell's to-do list. Because these responses involve transcription, they take longer to occur but last much longer. This makes intracellular receptors the slow burn storytellers of the cell signaling universe. Once all the signaling drama settles down, the cell finally delivers a response. Some responses are subtle, like adjusting enzyme activity to fine-tune metabolism. Others are bold, such as changing gene expression or reorganizing the cytoskeleton, like a microscopic interior designer. Cells may also release hormones, neurotransmitters, or other molecules to continue the communication chain. Different cells respond differently even to the same signal, which is biology's way of keeping things interesting. Many responses help maintain homeostasis, ensuring the cell doesn't combust or melt into goo. Without proper responses, signaling would just be noisy molecular gossip. The fight or flight response is a classic example where epinephrine triggers GPCR pathways to mobilize glucose and sharpen your reflexes. Insulin signaling is another essential pathway that keeps your blood sugar from behaving chaotically. Growth factors activate RTKs to encourage cell division, which is great unless they get carried away. Neurons use rapid synaptic signaling to pass electrochemical messages faster than you can blink. Immune cells rely on signaling to coordinate attacks on pathogens, like miniature biochemical superheroes. Many pathways overlap or share components, turning signaling into a tangled web of molecular madness. Despite the complexity, these pathways keep organisms alive, functional, and occasionally caffeinated. Cell signaling is the grand coordinator that keeps the entire body operating in harmony, instead of dissolving into biological anarchy. It allows billions of cells to work together as one cooperative organism, rather than rather than competing like unsupervised toddlers. Signaling enables you to think, move, eat, breathe, and occasionally break into spontaneous dancing. It helps cells adapt to changing environments, ensuring survival in a universe that seems personally invested in causing stress. When signaling breaks down, diseases like diabetes, autoimmune disorders, and cancer appear like unwelcome guests. Understanding signaling lets scientists design targeted therapies that fix communication problems at the molecular level. In short, cell signaling keeps life running smoothly and keeps biologists employed. That's it for now. Don't forget to subscribe, like, and check out Dr. B's tutorials on Amazon for full AP and IB study guides. See the first link in the comment below. For more tutorials and test prep videos, subscribe to our YouTube channel, Dr. B's Tutorials.